Welcome to the Potomac Institute for Policy Studies and our seminar that we're calling Meteoric Impact, Influence of Space Technology on Terrestrial Medical Advances. We're very pleased that you could join us this afternoon on this important conversation about the impact that our spaceflight programs have had on terrestrial medicine. My name is Curtis Pearson. I'm a vice president here at the Potomac Institute. And I'm sitting next to Dr. Alyssa Adcock, who is a research fellow here at the Institute, as well as the lead for this project.
Thank you, Curtis. It's truly a pleasure to be a part of this seminar today. We are so lucky to be joined by our three fantastic panelists to really explore how innovations and spin-off technologies from space exploration have advanced medical capabilities here on Earth and really highlight a lot of promising areas of potential future development. Space technologies and capabilities have left a profound impression on terrestrial life. Innovations spurred by space exploration have seen application beyond human spaceflight, addressing health and medical capabilities on Earth. From medical devices and novel materials already in use, to the possibility of entirely printed human organs as large as your lungs or as small as your eye retinas. The benefits that society and economies can reap from space exploration technologies have just barely scratched the surface. As agencies look to send humans back to the moon, to Mars, and hopefully one day beyond, society will continue to realize benefits from new space technologies and research. However, as space exploration has grown and reemerged as a prominent subject of discussion, so too has the debate on whether the cost is worth the outcomes. The value of space exploration, though, is recognized globally in countries such as Ethiopia, currently developing a space program, as well as the United Kingdom's National Health Service, partnering with the UK Space Agency and the European Space Agency in a multi-million-pound joint initiative. Stakeholders like this recognize the incredible innovation for terrestrial technology that space ventures provide. And so should anyone that uses an insulin pump, has gotten an MRI, had a virtual medical appointment, probably sometime this year, or even wears a fitness tracker like myself.
Thank you, Alyssa. Let me give a few words on how things will go today. We will have some opening comments and remarks by each of our three experts. Once they have finished those remarks, we'll open up the floor to some questions, both from Dr. Adcock as well as from those of you that are participating in this. So please use the chat function to put your questions in, and we'll try to get to as many of those as we have time for today.
Great. So to get us started, let me first introduce the panelists in the order that they will speak today. So we are lucky to be joined by Dr. Kristen Faub, who is an assistant professor at Baylor College of Medicine and the former chief scientist for TRISH, NASA's Translational Research Institute for Space Health, which is a high-risk, high-reward research and technology venture for NASA. She is also the incoming deputy chief scientist for NASA's Human Research Program, or HRP, which is dedicated to discovering the best methods and technologies to support safe and productive human space travel. Following Dr. Faub will be Dr. Christopher Mason, who is a professor in the Department of Physiology and Biophysics at Cornell Medicine. Dr. Mason's laboratory builds and deploys biochemical and computational technologies to understand and engineer human and other genomes. He is either a principal investigator, PI, or co-investigator on a variety of NASA missions as well as projects. Some of these missions are looking to discern the molecular basis of changes in the human body during long-term human space travel, including everything from genetic to epigenetic, transcriptional, and metagenomic changes. Following will be Dr. Ashvin Beheshti, who is a bioinformatician and principal investigator at NASA Ames Research Center, also part of KBR and the Space Biosciences Research Branch there. His research includes exploring how microRNAs will affect space biology and potential use for countermeasures to mitigate space radiation and effects of microgravity. Dr. Beheshti also holds a visiting research appointment at the Broad Institute of MIT and Harvard, and is the lead on a nonprofit formed last March in 2020, working on COVID, called the COVID-19 International Research Team. So to get us started, we also want to note that we'll be providing captions for this seminar. Instructions on how to use or turn on these instructions will be provided in the chat. This event is also going to be recorded and will be posted on the Potomac Institute's website for later viewing. So first, let's get started with Dr. Fobb.
Thanks for the background, and I'm looking forward to this discussion and certainly listening to what Asheen and Chris has to say following me. So yes, my background, I was a former chief scientist at the Translational Research Institute for Space Health. And so really thinking about how we're using the technologies of today, but also what we're going to do for the technologies of tomorrow for space exploration. So we'll talk a little bit about that. Also, just a little bit more background. I was at the NIH for a period of time doing my postdoc work and then going into program management on the Organs on Chips program, the microphysiological systems. Not sure how many people are familiar with it. It's fairly common now, but still it's tissue engineering is still somewhat new-ish. Then I also had a little bit of experience in the pharmaceutical industry as well. And so by being able to understand a little bit of an academia and industry and government perspective, I think that what's going to be clear from these discussions and how we're going to really make innovation drive for both space health and terrestrial applications are going to be a lot of partnerships within the agencies, but also public-private partnerships. So thinking about space exploration, so whether we're going to the moon, whether we're going to Mars or beyond, why are we investing in that? And so of course we could talk about philosophical discussions on why we do space exploration. We could probably do that all evening long. But for the context of this, we really want to start thinking about the technologies that are going to be needed to keep people safe, healthy, happy, sane during space exploration, and how that actually is going to apply to on-Earth applications. And really, in my former life at Trish, and what I plan to bring onto the Human Research Program and what they already think is, how does this apply and how does this relate to you? How does this relate to the individual? How does this help the community here on Earth? And thinking about the austere environment that space provides, it really does demand innovation to occur in order for us to really buy down these hazards and risks that humans would be exposed to in space. And so whether that's extended periods of time in microgravity, how your body will shift to microgravity, so the fluid that we're made of, thinking about the shape of our 3D organs, the scaffold changes that happens to our organs. And so thinking about what happens physiologically to our body in microgravity, what happens when we're in a confined environment. We're not exposed to new things. We're not exposed to new microbiome or to new people or new plants or vegetables. We're in this container for a period of time. What does that do to our immune system? What does that do to the microbiome? What does that do to the ability for us to have good quality nutrients as we're going through space missions? What does that do to our behavior? What does that do to our mood? And how these all connect together. So there's a lot of challenges that we have to think about, the types of technologies and methods that we need to make sure we have in hand are absolutely going to help on Earth. So how would that help on Earth? So I think that there's obvious examples. And so a lot of what we think about with radiation countermeasures or being able to have point of care can apply to military applications or keeping our troops safe. We can think about underdeveloped countries, and so developing countries, how we can provide medical access to them. But even a little bit more personal and more here at home, how do we handle things in rural communities? How do we handle unknown surprises like the pandemic that recently happened with COVID? And I think we'll have some examples of that coming later on after I'm done talking. So we need to make sure that as we are thinking about space exploration, we're preparing tools for the unknown, but those unknown tools can actually apply for today. And so providing these to easier medical access to developing countries or in rural environments, people that maybe not able to drive, maybe can't make it to their doctor very easily. Can we provide them with the tools in their house and so that they can have good quality information about how to handle their prognosis, their situation, their disease or their syndrome, or just their health and nutrition overall. So giving the tools for space, the crew, but for here on Earth, the patient, the population, to make sure that they have well-informed information to help be a part of the decisions that they want to be part of for their health. So that can be medical devices, being able to expand on capabilities and taking a drop of blood and being able to understand your organ function. Is your liver functioning okay? Your kidney values okay? Being able to have this readout at the touch of in the palm of your hand and the touch of a button, and being able to understand what those readouts are in an easy way. So that actually takes a lot of thought in engineering and biology in order for something that simple to be successful. So there's a lot of those kinds of situations where it could be medical diagnostics, it could be therapeutics or drugs. We can't take an entire medicine cabinet with us to the moon or to Mars. So we have to think about how we could create drugs on the fly in an emergency, in an unknown medical scenario. How are we going to be able to be fully resilient on that? So we think about synthetic biology. We think about how to use microbes maybe to help produce therapeutics, or how to use nucleic acid therapies, things that could be easier for us to transport and to create on the fly as we're on our way to the moon, on our way to Mars, but how that can actually help with point of care here on Earth, easier access to these types of technologies. So I'm really looking forward to what Chris is going to have to say and what Ashini is going to have to say, and hopefully this is setting the stage for using your imagination on what types of tools are going to be out there for both space health and for here on Earth. So I'll leave it at that, and then we can start off those discussions.
Great. Do you want me to jump in, or I guess I can just go now? Great. Oh, great. I'm being spotlighted. Great. If I'm on stage, sometimes it's dangerous, dancing or something. But anyway, really great. Honor to be here. Thanks so much for having me. And Dr. Fiber has really great summary, I think, of where the field has been and where it's going. Also, congratulations on the new position. That's should be very exciting. I think I saw some other announcements of lots of removing of departments at NASA, so I have no idea what that means. For you, hopefully it's just more resources, I'm hoping, or something. But and get to work with Craig Kundro as well, as an old collaborator on the twin study. So I think we're really at as most of you probably know, really kind of some people are calling it a space race 2.0, or really certainly a uniquely different phase of space exploration, certainly space biology, and also aerospace medicine than really anything that's come before. I think we have really a mirror of what we see for terrestrial medicine, or just what we do in hospitals, is that there too, in the last 10 years, it's really gone from much more of a responsive medicine to more proactive, and also personalized and precision medicine. And, a lot of the work we do in aerospace medicine is taking as money, it's a virtuous cycle, and really where the tools and technology to deploy in hospitals, we get them up to space, but then space has very limited resources, limited space, as you just heard about, and it forces you to innovate and have very small memory footprints for algorithms, and limited or fewer as few steps as possible for any protocols that you do in space. And so I think some recent examples just that we've worked with have been getting a really multi-omic and complete molecular and behavioral profile of astronauts, which had been done to a degree. There's a longitudinal health assessment study that NASA has been leading for decades. But there's also, a much more sensitive ways to ask some of the questions that have come up since the space program started, which is, what are the long-term risks for astronauts, if any, and if there's any, are they different than, say, more just basic risks that you might have for other jobs on Earth? Whereas before we, try and look at longitudinal assessment of health and incidence, say, of cancer or heart disease or general disease that would occur on Earth. But now we have, due through a lot of innovations in genomics, we can see very fine-grained resolution of mutations that are occurring at low abundance in stem cell populations. We can look at movements of how epigenetics shifts over time might indicate the degree of aging of cells or even stem cell function, mitochondrial function, which I'm sure Dr. Beheshti will talk about a little bit. We've published a lot on that. There's all this, there's a really extraordinary appreciation for things that just we didn't even really know about until 10 or 15 years ago. The very first papers were coming out. Some examples include assays to measure cell-free DNA, where we can actually look at nucleic acids in the bloodstream as a sort of full body molecular scan of what's happening in the body. And it's because every time you see fragments of DNA in your blood, it's there because cells have lysed or they have been excreted in, say, exosomes. It's like the body is a continual, well, it's a big fluid. As we heard, the fluid shifts are really pronounced in space, but really there's a lot of fluids that move around, but they carry so much information because every fragment of DNA not only tells you if you have cells that are dying, but also which cells are dying based on their epigenetic signature. When you look in urine, you of course can look for a UTI or an infection, but there too you can take any fragments of DNA and infer the tissue of origin just based on the DNA itself. And so again, I'm a geneticist, so I'm thinking of all these things about DNA and RNA applications. That's my obvious bias. I think genetics is the best though, but of course I think that because I'm a geneticist. But it's also because it has transformed very many aspects of medicine, I think, that most people are familiar with. Almost all non-invasive prenatal testing is done with cell-free DNA, for example. You no longer do amniocentesis or chorionic villi sampling. It's just a blood draw. And the same thing is true if you look at, say, cancer, tracing and monitoring. You can do it all just with a blood draw. So GRAIL is one of the most notable studies trying to do this proactively for the large patient population of 150,000 people in the UK National Health System. The idea is, well, what if we can measure all these things in blood that we couldn't really see 10 years ago? Can we catch something before it gets too bad? Can we catch a stage 1 cancer before it's stage 3 or 4? And the answer is very much yes. It's going to depend how well for different cancers. But I think deploying some of these tools, as we've started to do for some of the astronauts with NASA and are starting to do more even with private astronauts, like the Inspiration 4 mission that just launched, really opens up this new opportunity to get a lot more high resolution measures of all aspects of physiology. But it also opens up a chance to discover things that might not be relevant to health, right? So you might find all these mutations that are just passive. You might find other molecular changes that maybe don't matter. And so what's good is there's a lot of baseline data being collected for thousands, and this point now millions of people. So we'll have, I think, tools to address that question. But in many cases, the best way to address it is the monitoring of the astronauts well before flight, during, and then after flight, using some of these tools, and then being very careful with the interpretation. Anyway, that's just on the monitoring side. I want to say it's being, looking at cancer and even just fetal health. They are now standard of care in hospitals, and I think that they're beginning to be used for astronauts. But there's other cool technology that we've been involved with, the Biomolecular Sequencer mission with Aaron Burton and Sarah Castor-Wallace, which showed you can do sequencing in space. Back with Kate Rubins in 2017, it couldn't have been a more perfect astronaut to go up for that mission. She's a trained virologist and had her own genomics lab and virology lab. So, and she may even be, she announces I'm one of the short list to be one of the astronauts to go to the moon. I really hope she goes, because then she'd be the perfect scientist to go to the moon, in my opinion. But it's just because I'd love to see someone with a nanopore sequencer on the moon, like looking back at the Earth. It'd be a pretty cool image, I think. And those are tools, again, that were only developed in the past 7-8 years, that now it's flight validated hardware. If you want to propose something to do sequencing on the space station, you can just include that in your proposal, and it's not a question of flight validation, it's already there. So there's a lot of cool technologies that enable rapid sequencing from a genetics perspective. But I think, as Dr. Fabre mentioned, is other ideas of things like tricorders or scanners. There's the butterfly, basically ultrasound imager, was just flown on the Inspiration 4 mission a week and a half ago, and did great up there. So we're actually beginning to get some of that data back. And I think starting to think of more point of care, rapid testing devices. It's really an exciting time, because they were really science fiction only, again, 5-10 years ago, and now they're being flown and they're up in space, for at least for the pilot purposes. And that'll help things at places where, you have limited resources. If you were trying to sequence with a tiny sequencer and an iPhone, you want to be able to do that, during an outbreak in low resource settings, and that's possible. If you want to do a scan for any changes in vasculature or anything with ultrasound, that's now possible. So I think this is really exciting miniaturization on the diagnostics. And then one really last thing I'll comment on that was brought up earlier is the idea of synthesis to in flight or modification of cells potentially. Could you have probiotics that could help fix problems if you have gastrointestinal problems? Or could you can synthesize DNA on very small machines about the size of a toaster. Could you make your own primers, your own even possible therapeutics for long missions? Like as was just alluded to by Dr. Fabre, there's, at the moon or Mars especially, you'll have to make everything yourself. And so that was very difficult to imagine, I think 10 years ago, but there's now autosynthesis capacity for nucleic acids and a little bit for peptides. And I think that'll make it also easier for small molecules to be made. Essentially you have to bring your pharmacy with you. And that could be for making drugs that you might need, but also if you really did, actually, one of the interesting things that happened when we was describing some of the genetic results to Mark and Scott Kelly for one of the first genetics debriefs, and I just talked about this story in my book, so it's okay to talk about because Mark Kelly said it was okay, is that, one of the questions came, well, there's different mutations that have been observed and we think they're not meaningful. But one of the questions that came from Mark Kelly was, well, can you just CRISPR it away? Can you just fix the mutation that happened that may or may not have come from spaceflight? And the answer was, well, not yet, maybe someday, but certainly not anytime soon. It barely works in mice and there's a lot of off-target effects. So no, but it's interesting. I think there's this idea that's now in the public consciousness that cancer patients are getting customized T cells infused into them and people are having cells engineered for many purposes. And I was really surprised because that was on the face of it, a kind of a crazy question because the technology is not that good yet for it, but was also something that was fairly rational. If you've read the news and you think, oh, if CRISPR is so good, can't we just fix any problem that we have? And I said, oh, if only it was that easy. But I think that's still 10 or 20 years away. But I think it does raise questions of, where could we be 10 or 20 years from now? And that's I'll leave it a little bit with that sort of futuristic hypothetical. But that's what but again, it's based on everything we're seeing that we're doing now. There's over 1,000 clinical trials around the world modifying T cells and natural killer cells and then reinfusing them after they're grown up to target specific cancers and epitopes. And we're doing it routinely. There's 2 clinical trials at Cornell that we're doing this. So we know it's possible. It's just not quite prime time ready for astronauts yet, but it does raise interesting questions long term. And that's it.
No, thank you so much, Dr. Mason. So we're going to have our final panelist, Dr. Bichetti. I think we have some slides that we're going to pull out.
Yeah, I guess I'm breaking the mold here. I'm showing some slides and data.
Great.
But yeah, so everything that Dr. Mason and Fab said, I think it leads nicely to set me up, whether they knew it or not, to what I'm going to talk about. But yeah, just to go to the next slide, please. Yeah, so I'm going to be, kind of bringing it first with the space biology aspect of it, and like the research that, just from the examples that I do, for example, for research, other people, and actually Chris has been collaborating with these projects closely, and then how that you could actually benefit from that on Earth. So with terrestrial type of health effects and benefits, like Chris was talking about the cancer effects, and how that a lot of the research there could also be applied to space biology aspects. But one thing that I'm a physicist by training, I've got a PhD in physics, and then so naturally we're kind of like a jack of all trades but master of none. So that's sort of what we are. So when we joined, I switched over to biology and said, okay, well, they call us systems biologists. And so what is systems biology? And this is like a term I think was really started with National Institute of Health type research, where they realized biologists by themselves need all their disciplines, and same with all disciplines. We need all their disciplines. Like physicists need to work with biologists need to work with physicists, chemists, and so on. So the computational folks. This is so what systems biology is kind of one is looking at a top-down approach. So you're looking at a system as a whole, as the name says, and the other part is you instead of being in isolation in your own fields, working with all these new technologies that Chris was mentioning or Kristen was mentioning, and then bringing that together to actually understand how that might be a unified view of what things will happen. If you go to the next slide, please. So one aspect that that also is involved is computational modeling, which is heavily involved in system biology. So as you might have heard, like AI, artificial intelligence, machine learning, deep learning, it's kind of the basic definition of it. So artificial intelligence and things that you might think of, like let's say if you're watching Netflix, right? And the streaming, and how do they know what to recommend for you. That's sort of like machine learning techniques to decide based on all your patterns and thousands of other people's patterns. They could actually pick out, if you watch movies like Back to the Future, for example, and then Star Wars, well, then eventually start seeing a pattern from other folks watching the same type of movies. They can use machine learning techniques that way to predict, oh, maybe I could recommend that movie for you. But this kind of idea can also be applied to biology, and then space biology too. So, I'm going to show you some of that examples, how space biology techniques like that can be used to translate back to, what we do on Earth. So next slide. So one example, I'm going to talk about microRNAs. So that's just, you guys heard it was my background, and microRNAs kind of go nicely into like the fluids, as Kristen and Chris said. Basically we're all fluid, right? So microRNA is a really nice example of this. So next slide. Thank you. So what are microRNAs? So microRNAs are basically these small microRNAs. There's really small microRNAs, 22 nucleotides and things. And like the classical view in biology, as you see on the left side there, basically, we thought DNA would transcribe into RNA and then translate to proteins. So that's kind of the chain of events where, you would see the information happening. But it was 1993 when someone discovered, what these small RNA are not just debris. People usually just threw that away in the garbage because they thought it was just, fragmented RNA that didn't do anything. But then one smart person said, what is this? As a lot of, people might, one person's trash is another person's gold. In this case was truly is the case. So, what happened then, eventually in 2000, people started paying attention to it after the first person who looked at it in 93. There's a 70-year lag where people didn't believe anything that came out there. And then 2000, basically some people started discovering these small, tiny RNA called microRNA have lots of effects. And you can see the research exponentially going up for that. And the thing is, for Earth, this was all done terrestrial type research. So, the microRNAs themselves are in every disease related to good aspects and bad aspects. So cancers, there's a lot of microbes involved with development of cancer. There's a lot of microbes involved with good cardiovascular function, good health function. So, and it's pretty much, found in every organism, from plants to mammals to insects to even viruses, which I'll show. And then, the good thing about this is, you're saying about the fluids and the, fluid shifts. It's basically microRNAs are, because of their size, are very stable and they float around freely in your body. So then this will be kind of a nice thing for, let's say, biomarker development and assays, not only on Earth, but for space biology. So, and then it's basically the new understanding of it is, microRNAs are basically involved in every aspect. One microRNA can regulate thousands and thousands, or hundreds of thousands of genes. So it has a lot of influence. If you go to the next slide. So I'm talking about these little papers here. So you go on the next slide, we can just move on. So there's actually specific papers. So this is just a big general overview. I'm not going to go in the details of the paper. Chris was involved with this paper also. So basically the long story short is that, these microRNAs, using the techniques we developed, can actually then be used as a way to quantify your risk or your potential risk in spaceflight of health. So we basically use these microRNAs and we're able to show that there's a panel of microRNAs that are related to the damage caused by space radiation and microgravity events. Because as Chris alluded to, when you're in space, you got all these hazards, right? You got space radiation, you got microgravity, and then you can imagine space radiation is these big particles, heavy ions they're called, that go through your cells. As opposed to on Earth, you're protected by that, and you don't see that happen. And on Earth, the standard radiation is called gamma. So in a sense, you got the gamma is these sand particles that might go through your cells, and you're usually not exposed to that unless you go through, to go to a hospital, like an X-ray, or or any kind of standard things. And there's some background radiation that is not really too concerning at all, at really low doses. But you go to space, now you're exposed to those heavy elements, those big heavy ions that are like bowling balls, basically going through your cells. And so you could find different biology changing in your body. And one aspect of it was these microRNAs that were actually changing, and in turn, we found that it can not only can be this used as a good biomarker to indicate health risk, it can also be used potentially as a way to therapeutic, or what we like to say countermeasure, to prevent or mitigate these health risks. And one part of this was also, we looked at the twin study there, Scott and Mark Kelly, that Chris contributed, and we saw indeed that these microRNAs are really conserved and happening not only like in the mouse models and cell models we typically use in labs, but also in the human model. Okay, next slide, please. So then we started saying, can this be a potential therapeutic or countermeasure, as we call it, to prevent the damage? So it's not really therapeutic. So with another collaborator in Columbia, Pete, we have Pete Graham. We have he has like a 3D, as Kristen alluded to, there's like nice 3D microvasculature cell structures that people can make and use to simulate what can happen in a dish. So one thing he has developed is a 3D microvascular tissue structure, which is you see here on the top left. If you hit the forward button to go to the next thing. So, we had identified, as I said, a potential microRNA signature associated with the damage caused by the space environment. And this is what it was. And part of that component is associated with cardiovascular health risk. And since this tissue model is now associated with cardiovascular issues, this is why we chose those to actually potentially target. So let's go to the next forward. And you go to the next forward, because this is just showing you what potentially you could irradiate, you could simulate irradiations on Earth using high energy colliders and physics labs. And this is the Galactic cosmic ray simulated beam that we use. And this is like the composition of people who really wanted to know the details of that. But so what the images you see in the bottom is, you see that's the 3D tissue structure in the lower bottom of the panel C, 0 gray. So there's no radiation. That's how it's typically shown. So it forms this nice 3D vasculature structure. There's no blood flowing through there, but it's just the structure of it made out of endothelial cells. And indeed, and then when you actually irradiate these samples, as you see is the next panel there at the half a gray, that's the dose you might get if you go to Mars and come back, equivalent, like a total dose you get, an astronaut will get. You see that these vessels start collapsing, and you can see that the quantification is on the top right there, and the microRNAs are being overexpressed there. It says should be overexpressed with damage caused by the space environment. So you can see that happens next. Now, if you hit the forward button, that if you apply like an inhibitor, this is like an inhibitor that's associated with this, that can inhibit these 3 microRNAs that are being overexpressed due to the damage caused by space radiation. Would you be able to mitigate this response? So, this is through all the systems biology approach, which I skipped here because of our, just because I'm giving you a general overview of it. You could actually apply the systems biology technique to say, identify these microRNAs. We know it's a good biomarker now for health risk. Now, can we actually go to the next part and use it as a countermeasure to mitigate it? So we applied this, as the cardiovascular effect. If you hit the next slide button, and then, so this is just the background of it. So if you hit next again. Now, if you apply that, you would see definitely what happens. If my hypothesis holds true, you hopefully rescue that bit. And indeed, that's what happens. Only 24 hours before, again, this is a 3D tissue structure, cellular structure, and this is in vitro. So again, what we saw was that only 24 hours by applying this inhibitor, we completely rescued what we see here. So on the panel, you see the quantification of how the microvessels are formed and the quantification of the dimensions of it. So the collapse you see happen is the lower red box plot, and the blue now is with that countermeasure. We completely rescued it. And the last panel is just another control for the vehicle that you could deliver this without, the actual, it's like a scrambled version. So it's like another control, negative control, to make sure the actual vehicle doesn't contribute to rescuing anything, and it doesn't. So this is kind of an example of, showing what can be done in space. Like if you have kind of a toolbox, and and you do this to actually create a biomarker and a countermeasure, it might work. So can you apply this to something on Earth? So if you go to the next slide. So this is what it is. Now, can you translate the same model, the same blueprint, basically, and try something else? So now this is the COVID-19 research, which we've been doing this past year, doing the same idea. If you go to the next slide, thanks. So then we basically did the same thing. I'm maybe give a general overview, and also Chris was involved with this study too. And this is impressive. It was just recently accepted. So the preprint's available, but in a couple of weeks, you'll see the official publication, or a week or so. But the idea is the same thing. So, I said microRNAs, do they exist in viruses? Well, they do. And the thing is, the viruses and microRNAs have an interesting interplay where, you could actually, viruses can integrate some of the genome of the virus, as I show here, the overview. And then when it does that, it actually then does it for a few purposes. One is to actually then have a recognizable human component in there, because usually a virus, when it enters your body would say it's a foreign object, kick it out. But if it has a recognizable sequence that's a human component, it might get tricked. And that's what sort of SARS in former virus literature from like hepatitis C. That's exactly what they found in microRNA. Like this did that. And in that sense, once it does that, it evades the immune system, because the body's immune system gets tricked. And then what it does is then it overexpresses microRNA throughout the body. And once it does that, then it's sort of like napalm, recreates the landscape so the virus could survive. So then my hypothesis was this, when I was able to use the same bag of tricks as I used for space biology for COVID research. And indeed, like, using the bag of tricks that I did, I was able to identify microRNAs associated with SARS-CoV-2. And when we did further analysis of a lot of collaborators involved here, we found that indeed the specific microRNA is integrated into the SARS-CoV-2 genome. So does this actually lead to future, all the symptoms you'll see? And again, the same systems biology bag of tricks we use for the space biology work we use here. And we see indeed we could predict that it does. And so then we started actually quantifying this microRNA like we did for the space biology work in the actual patient samples from COVID. So we took, if you go to the next slide, there you go. Okay. So if we, took urine, serum, and nasal swabs from patients, COVID patients, this is what the slide is on the left side, and then quantified this specific microRNA, you see that it's just a general overview of it. You see that basically what happened is, indeed, in a COVID, for serum and urine, the fluids, as Chris was talking about, if the microRNA is, if it is a positive COVID patients, it's basically overexpressed in there and pretty much floating around in any of the blood or urine samples you see. While negative patients or negative COVID, even healthy patients, people who never had COVID or diagnosed at the time, they don't have this floating around. What's interesting is that the nasal swabs, from a nasal swab sample, nasopharyngeal swab sample, there's no difference. And the reasoning, or my thesis or hypothesis on this, would be that indeed, like this virus would express this microRNA once it's established in your system, and the nasal swabs just hasn't been established yet. So then once the virus gets in your body, then it starts floating around and then getting into tissues and causing the damage it does. And the microRNA will help it, rescape the land and get into your different organs. If we go to the next slide. So then they can use this, just as we did for the space biology work, can use it as a therapeutic. So this is now really preliminary, like this is in the paper, but this is really just the first run through of trying to see if you could inhibit this microRNA and does it have any effect. So this is, a potential antiviral, I call it, because, there's still a lot more work, like in investigational new drug studies you have to do actually get approved and see if it might not work, but, it has very, very good promise. So, when you do standard in vitro assay screenings that you do with collaborators, you would actually run these kind of assays for viral screens that you see. So with another collaborator, Nushri Chatterjee at she's a Sachi Biotech, small startup. She had this other form of self-delivery device. So we made this microRNA and then applied it to cells that were infected with the virus. And then we did a hamster study with another collaborator. And indeed, just long story short for this is that we see pretty good, zero-sided toxicity happening, but some really promising antiviral behavior happening with our inhibitor. As you would expect, you bind, again, just like the space biology work, you bind it, bind these microRNAs from preventing the happening, inhibit them, and then they'll stop, hopefully, propagating the events that you'll see. So if you go to the next slide. So those are kind of two examples of how, experimental models you can use to from space to terrestrial health. And if you go to the next one. So now, I talked about machine learning and AI. So there's other work that people can do. So one of my collaborators here, Diego Galeano from Paraguay, Ascension University there, he has this neural network model, which is, it's kind of fancy word for basically using machine learning AI technology to predict things. And so what his model is, can you use the nice algorithm, which I won't go into the details, but can you use a nice algorithm and find out if there's any alternative to target these microRNAs? And this is a more difficult task to do because right now there hasn't been any out there really to do that. And the point is, right now, microRNA, we heard from all the COVID work that others have done for the vaccines, right? That's mRNA, that's the whole RNA, right? So that's now becoming nice in the spotlight that, oh yeah, we can actually use RNA as a therapeutic or make vaccines, for example. But for microRNAs, so far, there's clinical trials that people are trying. So there's no actually proven track record, and there's a lot of legwork we have to do before we get to FDA approved places where we could actually do the therapeutics. So in the meantime, can you use these fancy machine learning algorithms and computational methods to actually predict drugs that would target these that are already approved by the FDA? And so this is what Diego has done. He's created an algorithm and a model and to look at first the space biology aspect of it. And does it, if you go to the next slide, this is a long story short, he was able to use his, if you go to the next slide, thanks. He basically can use his algorithm, for example, this is one of the microRNAs that were part of the space biology signature. And though he predicts a certain, based on his probability score he gets from his algorithm. This is some drugs that could be potentially used to target this microRNA. So then what happens? Then we will go back in the lab, test this against the microRNA, see if it inhibits it, and see if it does similar effects that you saw from the space biology aspect. Now he's done the same aspect, applied the same kind of algorithm for the COVID work. So now we have alternatives now to target that microRNA you saw from that, while we're trying to get a cleaner, the cleanest results to inhibit it like we're doing for the experiments. But in the meantime, we could use a more dirtier version by applying drugs or combination of drugs that are predicted to target these things. So that not only like the machine learning things are really applicable, where sure, we develop it for space biology techniques, but then it can be applied to cancer, in this case, COVID. In this case, like it could be many different things. You could use this model to target these microRNAs and then just run the experiments in the lab and have nice, never thought of before drugs that are already approved to target things. So if you go to the next slide. This one last machine learning like tools that people use. So that with other collaborators here, John Lawson and Ashwin from NASA Ames Research. So they have their machine learning tools that they use to actually look at proteins and how proteins can, let's say in this case, a cancer can actually then, like if you model the protein correctly, you can find how drugs can dock to them properly and find out the proper drugs that could actually dock to those proteins and the sites that might be optimal for it to bind to. So then in this case, you could find the optimal drugs that could actually, have the nice combination with the protein structure to actually be best, as opposed to blindly testing things that people sometimes do in the lab. So using machine learning tools to basically make it efficient to develop drugs. So initially this was used for space health, but then we applied for cancer. So if you go to the next slide, if you hit that movie button, if you hit play on the center, oh, no, go back to the next slide. Yep, if you hit play on there. So this is a simulation. Yep, this is a this is a simulation of some of their models, machine learning tools they use. This is like a protein model. This is for a specific molecule involved with cancer and lung cancer. And if you look at it, depending on what kind of profile the protein has, you see those helixes unravel, for example, this activation loop or the helix. And depending what state those things are in, that when it's activated, that's when you want the drug to come in and dock. And then depending on how that structure is, then you can use this tools to actually then design a drug to optimally dock to those places, let's say where the helix is unraveling or the activation loop's unraveling, to actually block it from activation. So this is like key understanding that applies not only, as Chris talked about, cancer, but you could actually then look at the protein level as opposed to the DNA and RNA level to come up with new small drugs that could actually, using these machine learning tools. So this is started in space biology, but also applies to cancer and other factors. And we're also applying this to microRNAs and other work. So that's kind of the basic idea. So if you go to the next slide, I think this is basically it. Yeah, so basically talking about how computational systems biology, computational model, is kind of a key way to understand space biology. But not only that, all these technologies, I think, as Alyssa had said in the beginning, like for example, your Fitbit watch came from space biology work. Some of this work, like for example, if COVID, the biomarker for microRNAs, or even as that antiviral works, that framework was set up in space biology work. So, there's a lot of crossover there. And then vice versa, as Chris brought up too, the cancer research going to space biology. And Kristen brought up all those other examples too. So there's a lot of things that go back and forth. Not only does, this terrestrial research help space biology, but it's the other way around too. So if you go to the next slide, I think that's one. Oh, and I would like to always make this plug that Chris and I and Susan Bailey from Colorado State put out this large package of cell papers, space biology papers that we organized with Cell Press. And one of them was mitochondrial factors that Chris mentioned too. So mitochondria is a big player in bioenergetics, it not only in space, but that impacts the space biology environment, impacts your body, but also mitochondrial, as you might know, is a big player for like any kind of disease. So because it's your bioenergetics, if your bioenergetics don't behave properly, then your health is going to decline, and certain diseases take advantage of that to make your health worse, because the disease likes to thrive off that. But if you want to learn more about a lot of space, like 29 papers in there from me, Chris, and a lot of other folks in the space biology community. So you can read about how those research and then you'll see the connections between a regular terrestrial research too. So that's it. Thanks.
One fun fact too, each one of those covers were pictures taken by Scott Kelly in space, so he got to pick his favorites, or really he sent them in the journal, pick their favorites, but still it's kind of fun bit of photos.
That's right. You can frame those.
Yeah.
Excellent. Thanks. Thank you to our speakers. Some incredible insights, which is which is why we do these forums to generate these discussions and so that we can learn about these incredible things that are going on. Everything from the idea that the austere environment of space requires innovation for us to understand and deliver care, and that can translate into better care delivery in underdeveloped countries or rural or very rural areas. The space flight developed monitoring systems that can then translate into early cancer detection and tracking, or these, as we just discussed, the microRNA signatures that help with the risks of space flight health that can then translate into therapeutics for both space and terrestrial applications. Just an incredible level of insights. Really appreciate your comments. Now I think we're gonna move to a Q&A section. And again, for those of you that have logged in, use the chat function if you have a question for one of our panelists.
Fantastic. So I'll start us off. I took so many notes. I'm really excited to have this discussion. So at the Potomac Institute, we're a think tank. So we do a lot of analysis of current and emerging technologies, but we do a lot of thought on what does the future hold. And Chris, you made some really great comments on reflecting back in maybe 5-10 years ago, how we didn't even have some of these capabilities. And so I wanted to spend first a moment to think through, well, what does the future look like? What can we imagine the future of both space medicine and healthcare in 5, 10, 50 years? And what does that look like then translating to terrestrial healthcare? So Chris, maybe you want to start us off with that.
Sure. I think, 5 and 10 years we can have some sense of, and 50 will be hard because it could be dramatically different, but that hasn't stopped me from talking or thinking about it. I just wrote a book recently about it as well, so why not? Let's go to 500 years while we're at it. So I think it gets much more vague as you go farther into the future, of course. But I think the near term is we'll see deployment, of some of these methods for monitoring and detection of any, physiological change, genetic change, begin to be deployed. And I think it's because it's already started, but we'll start to see more of it maybe monitored as part of what are called the standard measures that NASA has, or for private space missions. They just call them some of the biophysical metrics. There's basically this idea comes up again and again of there should be a minimal set of information we keep about everyone to gauge not only readiness for flight but safety after flight. And so I think more and more than we'll see deployed. I think in 10 years, if 10 years if all goes well, might have some of the first missions to Mars by 2035, 2037. It depends on, China might not be the United States. China might get there first, according to some announcements, but it's hard to know for sure. But it's, there is, like I said, a little bit of space race 2.0. It's not just countries racing, also private industry wants to get there fast. And of course, SpaceX has announced there'll be, hundreds and hundreds of people on Mars in relatively short order. But again, I think we'll wait and see. But all things being told, it's probably in the next 50 years. Like even maybe people won't get there in 10 years or 12, but maybe it's 18 or 20. But that's certainly all short of 50 years. So I think in 50 years, what's really exciting is we'll have as a really big challenge of having people have to do almost everything they can on their own on another planet, which is really an extraordinary concept, but is very close. I think so that means that everything have to be manufactured, monitored, and responded to as much as possible without any help or guidance from the home planet. So I think that's what I think is really exciting about 50 years. And then if you go to like longer, 500 years, I think you can go out to deeper into the solar system. You could start to look at, hopefully, what I propose in a recent book is that at the time we get to 500 years, we'd have enough knowledge of biology and safety deploying some of these tools that we could start to put people on generation ships that you could go towards an exoplanet that had been discovered. And there too, 20 years ago, we barely had a genome, and we had almost no exoplanets. You could count them on one hand, but now we have hundreds of likely habitable exoplanets, and we have a good grasp of genomics and medicine and even space medicine that's utilizing some of the same tools. So I think if you go away in the distance, maybe even we'd start looking at potential stars to head to, but that is a little bit, ahead of time.
Fantastic. I wanted to ask, Chris, you brought up the comment on some of these kind of autonomous systems, right? That they're we're going to have to be able to totally off planet without having Houston helping us out to maintain the health and medical care. And I know, Kristen, you touched on this a little bit in your opening remarks about this aspect of really these austere environments. I love the quote that space demands innovation. And so I wanted to see if you had more comments on not only just the impact that Mars or hopefully one day beyond mission will have on some of these rural communities or maybe third world countries, but also if we kind of flip that and what impact can places like you mentioned, the military and rural healthcare have then back for astronauts?
Yeah, I think that it's an interesting convergence of interests, I think, in terms of what federal agencies or entities are out there that are really starting to think about how does one benefit the other, and where can we use certain environments to help inform the other. So if we want to think about how can terrestrial environments help with space health, let's go back to something that, I touched on earlier, and so did Afshin talk about earlier, which is, 3D tissue engineered models. These have been around for, since the mid, like maybe around 2012, 2010. I think there were probably some basic models before that, but it really started to take off a few years back. And thinking about disease modeling and personalized modeling of these tissue engineered models, capitalizing on inducible pluripotent stem cells, where I could take Chris's cells and convert them into a cardiac tissue or to peripheral nerves or rudimentary brain tissue.
Brilliant, yeah.
And starting to understand, what makes Chris tick, what makes Ashvin tick, what would be, a disease model that we would be susceptible to. And if you would combine something with the power that Ashvin had, which is like systems biology, thinking about how we can start to take all of these different avatars of you, your tissue, your models, and have a you on a chip. And then being able to understand what diseases we would be susceptible to, and what, not only that, but what countermeasures, what therapeutics we would be really responsive to. That's already happening a little bit with the cancer that was mentioned earlier, how that can drive, okay, space isn't going to be just for that very small percent of people who are just genetically superior. I mean, they've got like the build, the intelligence, they're wonderful. I mean, they're people like me, who's like maybe not as genetically resilient to radiation, could be able to go to space because they can personalize and understand what risk I would be susceptible to. Helping make sure that I can be the resilient person that I need to be in order to go to space to do my mission. So the terms genetic superiority will then just vanish, that there is no genetic superiority because we can understand what risks are susceptible to everybody and what kind of countermeasures we can give to those people, both here on Earth and on spaceflight.
Yeah, no, fantastic. And I'm really glad you brought up kind of this aspect that we've been talking at the Institute for a couple of years with HRP. It's this aspect of precision medicine or precision health, right? It's tailoring the treatment, the diagnostics, the therapeutics, that proactive approach to healthcare. But underlying a lot of that is a conversation on data and being able to collect that data, but also understand it, analyze it, model it. And so I wanted to know, Ashvin, if you had some more comments on that, specifically of the role that a lot of this computational modeling and data is going to play into kind of the future of both medicine here on Earth, but also medicine in space.
Yeah, I think one for the space part I'll start with. I think one thing, like as Kristen said, is like, you need to have a pharmacy up there to, you can't drive up to your CVS or something from the moon and get, unless they build one up there, they might, but no. But so I mean, the idea is, so those machine learning techniques, to minimize the amount of stuff you bring up there, you could actually then predict, like you already know, let's say if you do a biomarker assay, like you're saying, you look at your fluids, oh, I have these factors up. So let's go back to my machine learning AI tool. Oh, you need this drug for that. So then you can really minimize, what you need. And that, of course, everyone's trying for that for the clinic, an AI approach, personalized approach, right? So if you have these certain symptoms, can the AI tell you exactly what's wrong with you, what medicines to take? And that's goes back and forth. People are developing that in the clinic. They're trying to. The computational biologists, they're doing some good work. Some really amazing work people are doing with like knowledge graphs and things like that, as it's called. But and that for the space biology, I think medicine is even better because I think was it in 2001 with Hal, sort of the same idea, right? You could actually create that, and you could ask Hal. Hopefully, he won't go crazy for our folks going to Mars, but as they did in 2001. But in this case, you make a more tame version of that, and they'll be able to predict. Okay, I have, for example, you take the blood, X factors are going up. If this inflammatory factors or immune factors are changing as you look at that. Then it can tell you right away what to do and what to make, and then you could minimize the impact of it, not only for payload, but also with drugs that you bring up there.
Yeah, it's there's Eric Topol wrote a lot about this, that medicine is personalized, precise, and there were and it's really tailored to your body, your physiology, and actually, yeah, Chris and I like the idea that there's no more genetic superiority. It's just everything's like a perfectly level playing field. You enhance some things that you want to increase or decrease the risks of others and make it so everyone has the greatest chance to go to as many planets as possible. It's almost like cellular. Every cell that you have could be everything it's ever wanted to be in some sense. That, but it has to be done. It does assume a lot that these things are robust, that they work in a really reliable fashion that could be really miniaturized. So a lot of the hardship coming ahead is to make things smaller, easier to use, more nimble, use less power. So that's the great thing about space, is it forces you to do those things, and it has all the benefits that accrue on the Earth as well.
No, but think about how what you just said, Chris, and bringing that to on-Earth applications. Me thinking about like the elderly population, where they might not have family that can take them to every doctor's appointment, and being able to have all of the tools in their house so that they can monitor and track, and that data gets sent to their doctors or team of doctors, where they can really be more informed about their health and make better decisions without the stress and the complications of going to these appointments in person, especially like during a pandemic.
Yeah, I was just not only the stress, but the risk. Like currently, the risk. A lot of people, people have resisted going to the hospital. Or actually, just had my cousin 2 nights ago. She was unfortunately got COVID. She's vaccinated, but got COVID as a breakthrough infection. She went to the hospital because she had been sick for 4 days straight. Couldn't hold food down. She got to the hospital and they said, "Well, I'm glad you're here." And she's actually just got infusion for antibody therapy. But they said, "Listen, we have no beds left. So if you want, you can go to the basement. That's our only option in the hospital." And she's like, "I'll just go home." So it's interesting that in a pandemic, especially, it's acute to realize that resources are limited. Hospitals are running out of space. You're like, well, if I could just get as much of that at home, including things like blood draws, like phlebotomy, you normally have to go somewhere, but there's a lot of devices. Actually, some are hopefully putting up into a parabolic flight in December, and then hopefully putting in the AX2 mission next summer is a Tasso device or drawbridge. These like really easy devices that just have suction, do the blood draw for you, stabilize it, and ship it. That way you don't have to, go to a high-risk hospital during a pandemic, or even just go to the hospital. On the one hand, I want to make it easier for people, but they should still move around. The good thing about your bodies were built to move, so you want to make sure that old people don't just do everything at home and never move. But so we have to make sure it's not too convenient. But it's really it's another one of the benefits. If you can make it easy for people to stay safe, to stay in the convenience of their own home, and then the physician's team and the physicians get all that information, hopefully in a HIPAA compliant, secure fashion, it's really enabling. It's really and it's kind of it's on the cusp of already being deployed. So yeah. It's exciting.
I guess one thing to add with the movement part, now AI part is time computational. As we already know, our Fitbits and phones tell us, hey, you stopped moving. So this is something, exactly. So this could be something like the AI technology, in addition to the health effects, they could say, as if we do everything from home, your phone will tell you to also move. But the astronauts, they have their exercise schedules, and they're told when to exercise, and it's all automated, right?
And so the red line, like the punishing red line on the ISS of what their tasks are, which they don't like, but they have to do it. But it is kind of interesting incentive. Like when I just have a Garmin, but like if it tells me I haven't run this week. I do feel like, you know, like I'm being guilted by an inanimate object. It's like, oh, I feel lazy because my watch does this number of how much I haven't run or something, but it works. I don't know if it, you know, it's actually quite effective.
Yeah.
Yeah, they're fantastic. I love this thought and future kind of thinking through also some of these like almost sci-fi aspects, right? You referenced some great movies as well, but relates to one of the questions that we actually had submitted through the Q&A on, are there limits to this? Are there financial limits? Are there scientific limits that we can speculate and project all of these things, but what are some of the limitations that are going to prohibit us from fully realizing some of these possibilities?
So I could maybe jump in first. I was to say the microRNA, that my limit is finance, because, you know, someone has to fund that, run the investigation drug studies, and that's like, you know, a couple of $1 million. And then once you get to that point, so like you might potentially have a cure or a therapeutic for something, but you'll never know until you apply for the grant cycle. You have to go through the grant cycle. Or you have some wealthy friends or friends of wealthy friends that can be like, you got to convince them. So, you know, you got to put on your used car salesman hat and try to be good at that. You know, so that the limitation, I think, for a lot of this stuff is money. You know, but if there's a hurdle that someone could actually say, oh no, you know, you could get over that. You know, that'd be that. At least for that part for me. And there's a whole bunch of other limits that Chris and Kristen probably know about that I won't cover.
I don't know. I could almost just go philosophical and just say the limits would be ourselves. You know, whether it's financial, political, prioritization. I mean, I do think that we need to like stop every now and again and ask ourselves some ethical questions, and see, you know, making sure that, you know, what the tools are that we are building are, you know, ethical and safe for the humanity. But really it's, I mean, we could say like current limitations, okay, like for today, I want to put tissue chips on the moon so that we can understand a real true space environment. You know, the limitations there are just that the technology just hasn't caught up, the engineering hasn't caught up. And so being able to build partnerships so that you can automate your chips and making sure that you can have these engineered tissues alive for 6 months to 2 years. And being able to pull up from your cell phone, like, I want to look at my cardiac tissues today. How do they look? I mean, I think that, you know, the building blocks are there. It's just the amount of investment to assemble it and to have partnerships to work together to get it there. But that's not necessarily a limitation. It's just more of like, you know, creating a strategy and a roadmap to get us to that place.
I think, yeah, definitely would agree with the structural, you know, financial limitations, the engineering limitations are still on the table. But I actually think some of it is a value or philosophical, you know, drivers, maybe the biggest one, because, you know, the amount of you know, it's like tenfold less of the United States GDP is for NASA these days, and that it's much, you know, but we're still probably never more has been asked of NASA, maybe to like going to not just the moon but to Mars, right? And like, we'll give you less budget, like a tenth of it's kind of crazy, just as a concept. Let's do the much harder thing. It's much farther away, much riskier, with much less money. You know, but academics in general are kind of used to that. I really like your grant, but we're going to cut the budget in half and then tell you to do everything ×2. So it forces you to be scrappy. But I think if we really, I mean, actually, this may be a bit of a sidebar, but sometimes I'll like tweet or post on Facebook about projects we're doing or papers that are published about, you know, aerospace medicine. And occasionally someone will say, well, you know, that's cool, but, and I think a lot of people in space research get this a lot, like, oh, that's cool, but, you know, why don't you study COVID or cancer, or like something that's more immediate is some of the concerns. Or one of them is like, well, why don't you study, okay, can you fix climate change? And there, I actually, you know, from a number of studies, so I'll often write back like, well, you know, climate change in a lot of ways and is a political issue. It's not really a technical issue. Like, we know literally if you just planted enough trees, would probably solve the problem in about a year. Like, so then that's not even that, like, it's a very easy, there's multiple papers last year that published that showed that. So like, I write back like I want to study science problems that are hard, that we don't know the answer to. Like, we actually kind of know what's causing it and we could immediately address it. For like, just trees, frankly, would solve most of the climate problem probably. But I think it's a harder problem, but it's, you know, it brings out the best in humanity for things we don't yet know the answer to. And I think people still have a value judgment sometimes that say, well, that's kind of esoteric, and, you know, there's more immediate questions. But I think that is one of the points that NASA makes all the time, other space agencies, like when you solve problems in space, it at the same time solves problems on Earth. And so I think it's just good to keep reminding people of that. Otherwise they'll forget that.
No, that's fantastic. And I'm glad you bring up the kind of global nature of human space exploration, right? It's not just one individual or one country, or right now, even just one organization, right? Where we're seeing that across the public, the private sectors as well. And so I wanted to also segue then into kind of what role does NASA and other organizations like ESA have to fill within kind of this sphere of advancing a lot of these medical capabilities, and what role or potential partnerships are there to have with organizations like SpaceX or Virgin Galactic?
We started some, I'll jump for, there are some resources already being developed where it's the private space agencies are working with those that are more government funded and academic. And so we've been working a lot with SpaceX and Axiom in the past, 6 months on the Inspiration 4 mission most recently, but a lot of other missions being planned. And the goal there is to try and share as much of that data with anyone that we can. What's interesting is, like beginning back to the barriers questions, one of the barriers is China. Like NASA is congressionally limited from doing anything with China, including sharing data, methods, materials, which I think, I understand politically why that is, but I think it's scientifically, unfortunate. It was very, even the year-long missions that were planned between Russia and the US, I had a lot of data sharing planned. Not as much happened as we wanted, but even there, what was a previously sworn enemy has been very collaborative, even during the height of the Cold War. So I'm hoping there'll be some way that politically it would be feasible to have more collaboration also with China, but I realize no one from NASA probably can even say that. But I mean, it's just too politically complicated, and it's congressionally driven, right? So you can't really, as much as we would like to, that might be hard, but it's kind of the third rail of space medicine and space research is how do we all work together more, including China. But it's easy for me to say that because I don't have any government appointment, so I can just say whatever I want and get everyone upset. But I still would like it to be true. We're all humans are my species. I like them. I like all the other species too, but generally humans a lot.
I really can't speak really for NASA yet, since I'm not officially on board. But what I would say, just from my experience, both in the private and public sector, is that there are strengths that a federal government agency has, and there are strengths that private entities have in industry. And so while we can think of government to be big and bureaucratic and have a lot of red tape, they also produce a lot of good science and invest in a lot of good science that's available to the public. So therefore, the knowledge, the research, the missions are for the public good. Whereas industry, it's usually about the company, benefiting the company and growing it. You might be building a technology like or a medical device or a drug, but often these are for-profit entities. And so what I think that they can strive in is faster development, being able to hire a lot of talent to drive things farther, quicker. But they don't have the necessarily the strict or the restrictions that a federal agency can have. So they can be a little bit more agile. So really, I think that it's going to take a lot, going back to what I said earlier, the strengths of the private sector and the strengths of the public sector coming together to really get us to be successful with a combined budget to get us to a safe, more realistic day-to-day space exploration life, the space 2.0, as Chris said. So I think that we really just have to draw on the unique strengths that both the private and the public sector will give.
Yeah, I couldn't agree more. And actually, the same thing is true with like development of any terrestrial medicines, terrestrial applications. Like if you want to do something kind of tinkering and very fundamental, often it's like in academic labs. But then if you want to scale up a platform, or if you want to manufacture 1 billion vaccines, it's not going to be done at Cornell or in one person's lab or even 5 people's labs. It's a large scale manufacturing question. So there's differences in speed and scale, but I think having both is key, and having them work together is even better. Yeah, I couldn't agree more.
And I think one thing that is key too, as Kristen and Chris brings up, is the collaborative networks. Like, yeah, I think a lot of folks fall in that habit. I know Chris doesn't. He has a large collaborative network, and Kristen, of course, you know everyone because you've been involved in Trish and all that. So that's good. But so we're all on the same page. But the thing is, like a lot of classic labs, they isolate, they stick in their lab, or even industry, like they stick there. We can't share anything with anyone because we're going to get scooped, because that goes to the funding part. But if everyone has an open science idea, which I know Chris does, and Kristen obviously supports open science, I would hope, I think, talking to you, that we all think that things get done faster. And we see that with the COVID pandemic. You see a lot of the good papers have 50 authors or more. I'm coming from a high energy physics background, and all our papers are 100 authors alphabetically ordered, and everyone knew what they did, and everyone gave credit outside. So that's fine. But that's I think that's the new way. If industry and academics is what Kristen and Chris was saying, they come together, because then you have collaborators all the way in Japan, for example, we have Australia, or South America, all over the world. And nowadays everyone's Zoom, so it doesn't matter. You just have to stay up a little later or get up a little earlier to talk to those folks, right? And that's all it is. And I've noticed, like the COVID and with space biology, it's my same attitude. Just get 50 people aboard, and you get things done so much quicker. And this is the way forward. And I think industry is seeing that too. So I've been hearing a lot more from industry folks, at least outside of the U.S., from Japan and other folks are like, oh, I see capabilities here in the U.S. that we can, so let's collaborate with the academic folks in the U.S. So I think that's the next 10, 15 years. I think that should happen. I don't know if that how many folks will be on board of that, but maybe COVID has changed the attitude of folks.
I think that that has changed a little bit, and Ashton, you've seen this from me at Trish, or the previous agency that I was at, is like encouraging collaborations. And so understanding your portfolio, if you're a program scientist, and understanding the strengths of each project that your institute is funding, and being able to help connect dots, and encouraging conversations with the PIs to be like, hey, I really think that Chatterjee's Phasmers would be really useful to your mRNA project with COVID. I think it's a culture change, and being able to use all of the tools, whether it's in industry or public, just to be able to say, hey, there could be more money if we're partnering together, and your projects will go farther if you guys can work together and are able to work together.
And that's why I appreciate it from what you've done, that you facilitated that. I think that's what made it so successful, that program you were in charge of. No, it's true, because you said, hey, you people should get together, and you kind of guided people that I don't think initially would have come together. I think that's key.
And I see that as not an outlier in the future. I see that becoming the norm, especially as budgets shrink. Being able to be clever about the resources and the areas of investment and how to partner together.
Mm-hmm. And samples are limited too. Any of the biospecimen samples for study, they're always limited, so you kind of have to. Yeah. There are some questions in the Q&A. Do you want us to jump in on some of them, or do you want to pick them?
Start with a couple of them. One that I thought was an interesting kind of discussion, and I think Kristen, you kind of brought us to the bioethics aspects, and so I wanted to pose the question: what negative implications maybe have the potential to arise that we hope don't come to fruition or use? And I know, Chris, you had mentioned the aspect of just CRISPR it, and so I know there's been concerns in the community over
concerns, and well-validated ones. Henkei Ji already CRISPRed human embryos with an ostensibly reasonable idea of CCR5, if you have a deletion in that gene, you'll be immune from most strains of HIV, and that he thought about reaching out to families that had been suffering from HIV. So you think, well, okay, maybe that is not necessarily an awful idea, until you think about how good the technology is, and it's not quite that good because there's off-target editing, and the fact that CCR5 is pleiotropic, meaning it has more than one function like most genes do. So even though it would decrease your risk of HIV, it increases your risk for West Nile virus. And so, like most things, there's this balancing function in the genome, shaped by, in some cases, billions of years of evolution, if not hundreds of millions of years. And it's hard to just tweak them. So I think some of the negative implications are thinking we know what will happen if we start to change things, and then being wrong. And that's really the crux of systems biology and synthetic biology, is how good will your prediction be of what you change. But that's why you start with cell lines. You start with animal models. You do things in a way we've done clinical trials for decades. is we're going to do the same thing, I think, in space medicine. And so the one risk is going too far ahead before we know exactly what the technology is doing. And one place where there's a lot of new developments is in epigenetic CRISPR mechanisms. You can use a deactivated Cas9 that lands on a surface locus, but bring with it something that modifies histones or DNA. So you can just tweak gene expression rather than modifying DNA, which opens up a lot of new avenues, but also opens up ways to do things wrong. So I think it has to have a little bit of a humility while aspiring for some of the largest feats that humanity's ever hoped for. So it's an interesting balancing act, but I guess I want to make sure we go very slowly before we go into like human trials.
I mean, just kind of,
Yeah.
I mean, even building on what you were saying, and any time that you're, for lack of better terminology, just tinkering with the genome, the DNA, what does that do? What affecting something upstream here, what does it affect downstream here? I mean, we think that we know a lot about the cell, and I think we do, but we still have so much more to learn about how impacting this protein or this mechanism here impacts everything on the cellular, the tissue, the functional, the organism level. And so we have to be really careful about, I think this even kind of goes to a question asked earlier about biohacking. Is like, being able to hack the genome so easily, is there going to be negative effects to that? There's always biological warfare, and the more tools that we try to build for medicine, does it have a double-edged sword to it? And quite possibly it does. So I think that's why we need to continue to stop and pause when we think about the ethics, and to make sure that we might have a bit of ability or capability for fail-safe if things were to go awry. Yep. Something to consider.
Yeah, and even in gene drives, which I don't know if people in the audience have heard of them, but these are self-propagating genetic systems that could modify ecosystems. Their discussion is, could you have reversion drives that if something goes wrong, you're like, oh, we made a problem, we'll just send a second gene drive through, which sounds like every bad sci-fi movie or Jurassic Park beginning of ever that you could possibly imagine, right? Oh, we'll just do it on an island, it'll be okay, and then something goes wrong. So that's actually what's being done for a gene drive trial outside in Nantucket right now. It's being done on an island with Kevin Esfeld. So hopefully it goes well. But I think the tinkering and the biohacking, it's early, but people have already started. So I think, so that's the risky part, but the advantage part is if you look at, for example, beta thalassemia, the trials, there's a New England Journal of Medicine paper 4 months ago, and this extraordinary, someone's already had gene editing done in, basically in vivo, like in their body, somatic gene editing, beta thalassemia. So basically you turn off adult hemoglobin and turn back on fetal hemoglobin. And it's extraordinarily successful, basically completely cured a year out, as far as we can tell, fully functional, and has cured a disease, right? So it's still early, but there's extraordinary, amazing studies that have already shown great clinical efficacy. And I think hopefully we'll see more of that in the future. I'll put a link to the paper actually, and I just actually lectured to med students about this. So it was I gave a whole preaching story about what could be done, and then I ended it with what you could do wrong. So I did both. It was there.
I love it. I wanted to, in the same lens of these kind of potentially unintended consequences with stuff, I wanted to go back to some of the aspects on this artificial intelligence, machine learning aspect that Ashwin had mentioned earlier. And so kind of what are potentially some of those implications for a lot of these AI systems or predictive modeling? Are there potential ways this could negatively impact healthcare?
Yeah, I mean, as with anything, you got to be like, computer models, there's bad ones and good ones, right? Just like any other predictive tool. So, of course, if you got to be careful. There's a lot of models out there, and you got to be careful what you use. But I guess the negative impact. I mean, I'm a proponent of saying AI will always have positive impact. but I mean, of course, there's always negative impact if you look at the sci-fi movies and see what happens, like Terminator and things like that. But I don't think that's real. But the truth is, like negative impact could be, yeah, sure, you could have the model's only as good as your input data in, right? Because in machine learning, you can't predict stuff that isn't there, So let's say someone tries to use a machine learning technique to predict some disease that there's not that much literature on. You could get drugs predicted that might not be very accurate, you know, so or gear you to the wrong direction because that data in is garbage, the data out is going to be garbage. So depending on how careful people are, you know, that's the case. So that's the limitations when it comes. It's really, I think the machine learning tools are only as good until like the biology is limited. So that and then the biologists have to step up and say, all right, we'll create some data for you and parameters to use for your tools. You know, so it's like an interplay. You need both. But that's the thing. So, but some people might fully hop on machine learning and not really pay attention to that beginning input of the data.
Yeah, fantastic. So we have about 8 minutes left. So I wanted to give each of the panelists maybe 60, 80 seconds to kind of give some final thoughts. We're also going to leave, I think, the Q&A open after a little bit after our 2:30 mark. So if additional folks have more comments, we'll have a little bit of time for that. But I'll start the order that we started in this morning to Kristen, if you have some final thoughts.
Sure. I'm excited. I think this is a really exciting age to be in when it comes to innovation and research and technology, both for the types of research and the technology that is on the precipice of existing, and the diversity and the mixture of people and different perspectives that are starting to become more involved in STEM, and you know, what new perspectives are going to be coming to solve these problems within our next generation. I'm really excited about that. And I think that I'm excited to see how a lot of this is actually going to help the earth too. I mean, like not only that, just like positive environment, like being inspired again in the space race, inspired Americans to be Americans. And you know, being able to find these step change discoveries, I think could really be a healthy thing for our country and the world, really, the globally. I mean, speaking to like even just the last question here on climate change, you know, space, you have to think about sustainability. And you know, can we start to think about the technologies that we are building, you know, maybe using living biomaterials to help with our structures, our scaffolds? Can we apply that to the earth, and so that we can actually have a better climate, not just for our crew in space, but like how we're applying that to improve our climate here on earth. There's a lot of hope there. It's just people have to see it.
Yeah, thanks so much, Kristen. I agree with the hope. It's actually kind of a new hope. It's like Star Wars 4 or something. It's a new hope. You know, it's happening. There's a new kind of hope because you know, we really, you know, I think just to the point of climate change and the world we're living in, there is a sense of, you know, a sense of pride people have. I actually just went to the first launch I've ever been to was to the I-4 launch a couple of weeks ago, and it was, you know, just. It is so truly an awe-inspiring set of ships we've built, progress that we've made as a country, but also around the world. So I think it really is inspiring, even without just seeing it, but just seeing all the progress. And I also think that we, you know, in some point can seem daunting. Like we're trying to manage at a planetary scale our resources and environment and atmosphere, and get to other planets. But we could, I think, you know, and it seems like maybe we're not doing a good job because CO2 levels are still going up, species extinction is still increasing, habitat destruction. There's a lot of things you could bemoan, but I think, and appropriately be worried about, but I think it may have been like a growing pain we had to go through. I think to really address planetary scale challenges, we needed to reach an information age where we could actually communicate and coordinate at a planetary scale, and then pretty soon interplanetary scale. So I think, even though it seems in some people are pretty pessimistic about the future, I've never been more optimistic, and I think we probably couldn't have addressed things as quickly as we want on as large scale until we've reached this kind of information age. Although that being said, we did address the ozone layer problem, you know, in the 80s, and that was something that, you know, has been done and could be done again if needed. So I think, yeah, I still think there's lots and lots of ships pointing to the sky. And it's never been going faster, so it's pretty awesome. Yeah, there's quantifiably never been more ships that have been things flown into space in the last year. So lots of things going up and staying up, and also there's more genes being discovered. So there are 10,000 genes discovered in the past 10 years. So we're still discovering things left and right. So it's pretty fantastic.
Yeah. So I mean, I don't have much more to add other than that I think space in itself is sexy for everyone. It's bipartisan, right? Whether you're Republican, Democrat, Independent, whatever other affiliation you want to go to, everyone loves space. So I think that's the thing about the hope part that Kristen and Chris brings up, the new hope. As long as you don't have some evil emperor come along and try to take advantage of it, which we're not at the stage yet. But right now I think that's what I think it unifies everyone. So that's the only thing that I can actually see, on Earth, that no matter who you are, everyone loves it. So and then I guess the other question would be, why do we spend all our resources? I think we covered this, that that for 8 people to go, like 8 astronauts to go to Mars or something. But I think that that was the obvious question. That the point of this was that it shows all the benefits you could have from technology and health on Earth. But I think the main point is, I think the hope part is I think this kind of brings everyone together, and that's I think inspiration alone to keep doing space research.
Yeah, everyone likes to look to the stars.
No, fantastic. So I want to thank you all again to our 3 fantastic panelists and the audience that was able to join us. I think this has been an incredibly insightful and really engaging discussion. How we've highlighted not only how the innovations that space technologies has been able to apply for advancing medical capabilities on Earth, but also thinking about where this might lead us. What does the future look like? How do we get there? And then what are those implications when we potentially do get there, both positive, but also potentially negative, right? Thinking through the issue in total. So I want to say again, thank you on behalf of the Potomac Institute to the panelists for this really, I think, stimulating conversation. And then thank you to the audience for your thoughtful questions in the chat.
Thanks so much. It was fun.
It's a pleasure.
Yeah.
Thank you.
My thanks as well for both our speakers. This was some wonderful insights and the real reason behind why we do these forums. And thank you also to the attendees for joining in the conversation, for your very insightful questions as well. And just as a reminder to keep an eye out for our next forum, which by registering for this one, you will get email notification as our next forums get planned and happen. So look forward to seeing you all again, and thank you very much.